So hello all. So today we'll be talking about we'll be discussing the question papers. So question papers especially from the last 5 years of NEET PG. Okay. So without any delay, let's move on with the first question. So the first question here is, in low insulin glucagon ratio, which of the following is correct? So first let's analyze the question. So they've given that is there is low insulin glucagon ratio, which means insulin is less and glucagon is high. Okay, so when does such a scenario occur? So when insulin decreases, what does it mean? So basically, let's leave off this insulin glucagon stuff. Let's analyze what will happen in well-fed state and fasting state. So if you are feeding, okay, so if you're feeding, what happens to the blood glucose level? So the blood glucose level increases. Okay, so when blood glucose level increases, the body tries to move the glucose from the bloodstream into the cells, right? It tries to push glucose into the cells. So which is the major hormone determining this, or which is the major hormone which brings about this mechanism? That's nothing but your insulin. So insulin is the predominant hormone which pushes the glucose from within the bloodstream into the cells, right? So insulin level increases. So whenever you whenever you take in food, insulin level increases to push the glucose into the cell. So what happens when you're fasting? So when you're fasting, the blood level of glucose is very less, right? So what will happen to the insulin levels? Insulin levels will also drop down. Okay, so because insulin level drops down, which other hormone increases? It's your glucagon. So glucagon level increases. Fine. So this is happening in well-fed state and fasting state. So now the question given here is in low insulin glucagon ratio. So when in which of the two phases does insulin level increase and glucagon level increases? It's your fasting. So it's in fasting that insulin drops down and glucagon increases. So a person is fasting, which means that the blood glucose level is less. So the body tries to produce ATPs, produce energy, making use of some other substances. Okay, some other substances, as in by breaking down fats, by breaking down proteins, by breaking down glycogen reserves, and so on, right? So these are the things which happen. So in fasting, what all happens? First thing is glycogen breaks down. Okay, so glycogen breaks down, glycogen level decreases. Second thing that happens is gluconeogenesis happens, because it tries to produce glucose from other non-carbohydrate substrates. And third thing that happens is it tries to break down fat. So fatty acid oxidation occurs. Okay, fatty acid oxidation occurs. And fourth thing, as a last reserve, your proteins tend to get broken down. Fine. Okay, so now let's analyze the options given. So first option that they've given is increased activity of hormone sensitive lipase, and the second option is increased activity of lipoprotein lipase. Now we'll have to look at what is the function of hormone sensitive lipase and what the function of lipoprotein lipase is all about. So now let's see what is HSL. So HSL, as the name indicates, it is sensitive to hormone. Which hormone? Nothing but your hero here, which is insulin. So whenever insulin level increases, the HSL level drops. Okay, so they both have an inverse relationship. Fine. So so what happens in fasting conditions? So the scenario given here is with that of fasting. So in fasting, what will happen to insulin levels? Insulin levels will decrease. So when insulin level decreases, what should happen to hormone sensitive lipase? It should increase. And what is the function of hormone sensitive lipase? See, basically the glucose level in blood. So imagine this is your bloodstream. Okay, this is your bloodstream. So glucose level is very less. So the body tries to give ATPs by breaking down some other substances. For example, let's consider this is your muscle tissue. Okay, so we have heart, adipose tissue, and muscles in which triglycerides are stored, right? So these triglycerides are nothing but it's a combination of fatty acid plus glycerol, right? So what happens is this HSL, hormone sensitive lipase, which is present in the adipose tissue. Okay, so the adipose tissue has HSL. Okay, so this HSL, so we have muscle, adipose tissue, and heart, which has these triglyceride reserves. So imagine this HSL is sensing that there is low glucose in the blood. Okay, so now what it happens is it mobilizes the triglycerides from the adipose tissue, That is, after breaking the triglycerides into fatty acids and glycerol, it mobilizes this fatty acids into the bloodstream. Okay, so now there is increased fatty acid in the blood. Okay, so this fatty acid acts as a source of fuel, okay, source of ATP. So this is the function of HSL. Okay, so when insulin level drops, HSL level increases, it increases the fatty acid levels in the blood, which will then act as a source of fuel. Now talking about the next thing that they had given, that is lipoprotein lipase. So LPL is nothing but lipoprotein lipase. So the function of lipoprotein lipase is just opposite to that of HSL. Basically, so if HSL level increases with decrease in insulin, what will happen to lipoprotein lipase? So lipoprotein lipase levels decrease with decrease in insulin. They both have a direct relationship. Okay, so they both have a direct relationship. And what is the function of lipoprotein lipase? So the function of lipoprotein lipase is, imagine this is your bloodstream. So there is fatty acid in the blood. There is more fatty acid in the bloodstream. So it mobilizes the fatty acids into your muscles, adipose tissues, and heart muscles. Okay, so it simply mobilizes the fatty acids and it is stored in the form of triglycerides. Fine, so that is the function of lipoprotein lipase. Fine, so next is glycogenesis. As already said, glycogenolysis is the one which is happening in such a given situation. So they have said that there is increased glycogenesis, which is again not true, and increased glycolysis. As we all know, glycolysis is something which happens as the first thing as soon as you take in food. Okay, so it is something which is happening in well-fed state. So this is not true, this is not true. Increased activity of LPL, again it is not true because it is low insulin level. So what is the right answer here? So the right answer is increased activity of hormone sensitive lipase. Fine, so this is it. So now let's move on to the second question here. The second question here is, okay, a chronic alcoholic had a high NADH-NAD ratio with normal urea. Which of the following is expected in this individual? Okay, so they've given the options as well. That one thing is low lactate, high glucose, high urate, and low urate. Okay, so first let's talk in brief about alcohol metabolism. So imagine the patient has taken ethanol. So what happens to this ethanol? Ethanol gets oxidized to acetaldehyde. Okay, so first thing is it gets oxidized to acetaldehyde. So the enzyme involved is alcohol dehydrogenase. Okay, alcohol dehydrogenase. And in this step, what happens is NAD gets converted to NADH. Okay, so what is the next step that is involved in alcohol metabolism? This acetaldehyde gets converted to acetic acid. It gets oxidized to acetic acid. And what happens in this? Again, there is NAD that is getting converted to NADH, and the enzyme here is aldehyde dehydrogenase. Okay, aldehyde dehydrogenase. Sorry. So what happens? NAD is getting converted to NADH. So what happens to the NADH-NAD ratio? It increases. NADH is more and NAD is less. Okay, so NADH is more. So what will happen to pyruvate? So initially, so when glycolysis sets in, we get pyruvate as a end product, right? So this pyruvate can have two fates. One thing is it can get converted to acetyl-CoA. Okay, so one thing is it can get converted to acetyl-CoA. The other thing is it can get converted to lactate. Okay, so which of the two pathways do you think will happen in a chronic alcoholic? So because pyruvate on getting converted to acetyl-CoA, what is the other thing that is happening here? So here what will happen is NADH is produced. Okay, NAD to NADH happens. But on the other hand, if pyruvate is getting converted to lactate, what is getting converted to what? It is NADH which gets converted to NAD. Okay, so which of these two arms do you think will be active in an alcoholic? Because already we have so much of NADH, right? So excess of NADH production will be futile. So the body will try to use up NADH. So this particular pathway will be predominantly active. Okay, so pyruvate getting converted to lactate is what is happening in an alcoholic because of high NADH-NAD ratio. Okay, so what happens as a result of increased lactate? So already we can rule out this option A here. Low lactate is not what is expected. It is high lactate. Okay, high glucose, no, glucose levels will also drop down because this thing sets in here. Okay, so because of high NADH-NAD ratio, glucose is also metabolized, and hence this is also not an option. So let's decide between high urate and low urate. What do you think? High urate or low urate? It is high urate. Why? Because uric acid, okay, uric acid and lactic acid, both these are acids, right? So both these things, they compete with one another for excretion in the renal tubules. Okay, so because lactic acid levels are higher, much more higher than uric acid levels, lactic acid outcompetes the uric acid in getting excreted. Okay, so this uric acid cannot get excreted efficiently, so they get accumulated in your bloodstream. Okay, so the answer here for the second question is high urate. Fine, so this is it. So we are done with 2 questions. Now let's move on to the third question. The third question here is, which one of the following is true about Warburg effect? Okay, so before going on to the options, let's discuss something about Warburg effect. So what do you mean by Warburg effect? So Warburg effect is something which is observed in cancer cells, right? So these cancer cells, what do you mean by cancer cells? Cancer cells is nothing but uncontrolled proliferation of cells, right? So the tumor cells, they go on rapidly proliferating. Fine, so these tumor cells, they rely on what? Aerobic glycolysis. Okay, is it true? Am I saying it right? Yeah, it's called as aerobic glycolysis, but then it ends up producing lactate. Okay, did you understand? So even though, when do you call aerobic? So whenever oxygen is present, we call it aerobic, right? Whenever oxygen is absent, then it is anaerobic. Okay, so remember this prefix here, aerobic or anaerobic, primarily applies to the presence or absence of oxygen, okay, and not on the fate of pyruvate. If pyruvate ends up getting converted to lactate, it does not mean it is an anaerobic glycolysis. Okay, remember this, pyruvate getting converted to lactate and pyruvate getting converted to acetyl-CoA. Normally, by convention, we all tend to use the term anaerobic glycolysis when pyruvate gets converted to lactate, right? So it's not the ideal way of saying so, because pyruvate getting converted to lactate or pyruvate getting converted to acetyl-CoA has got nothing to do with the prefix here, that is aerobic or anaerobic. So that the aerobic or anaerobic glycolysis, in order to determine that, we have to analyze, or we have to look at the oxygen levels. If oxygen is present, it is always aerobic regardless of the fate of pyruvate. Fine. Okay, so even in the presence of oxygen, tumor cells, they predominantly end up producing lactate more than acetyl-CoA. Why? What is the reason? Because anaerobic glycolysis, what happens, the ATPs produced are less. Okay, the number of ATPs produced are less, but the number of ATPs, what is the amount of ATPs produced by anaerobic glycolysis? It is 2 ATPs, right? So whatever ATPs, the number of ATPs that you get out of anaerobic glycolysis, that is like immediately available, right? It's an immediate source of energy rather than oxidative phosphorylation. What do you mean by oxidative phosphorylation? See, pyruvate eventually gets converted to acetyl-CoA, right? But yeah, although the number of ATPs that is produced by this mechanism is more, that is, you get 7 ATPs, right? Pyruvate to acetyl-CoA is always giving you, so this pathway will give you 7 ATPs, not pyruvate to acetyl-CoA. The entire aerobic glycolysis is going to give you 7 ATPs. But what happens is, this acetyl-CoA now has to enter into TCA cycle. Okay, so from TCA cycle, how many ATPs will you get? We'll get 10 ATPs, but it's not like, it is not direct. The 10 ATPs have to be derived from the reducing equivalents. So we get ample of reducing equivalents like NADH and FADH2, all those things, right? So these NADH and FADH2, they eventually have to enter into electron transport chain. Only after they enter into electron transport chain, excuse me. So only after they enter into electron transport chain, they'll be able to produce 10 ATPs. Okay, so that is why if oxidative phosphorylation sets in, ATP production will be delayed. But on the other hand, if pyruvate gets converted to lactate, ATP that is available is immediately available. Okay, and one more thing is, in cancer cells, glycolysis is not considered as a source of fuel. So glycolysis is process that is happening in tumor cells for the reason that it can be what to say, it can be used as a reserve of carbon skeletons, reserve of nucleotides, and as a reserve of even these things, your amino acids. Okay, so all the glycolytic intermediates, they get converted to nucleotides. Like glucose 6 phosphate can enter into your HMP shunt pathway, right? So from HMP shunt pathway we can get purines. We can get purines, so that way we can get nucleotides. And carbon, of course, the glucose moiety has 6 carbon in it, so all the carbon backbones, because a tumor cell has to rapidly divide, right? So it needs more of carbon and amino acids. So all these things are brought about or derived from glycolytic intermediates. Amino acids, especially, you have serine and glycine. Serine and glycine is obtained from 3-phosphoglycerate, which is an intermediate of glycolysis. So cancer cells, they utilize or they depend on glycolysis for these intermediates rather than ATP. Okay, so ATP is produced but in a limited amount than in a normal cell. Okay, in a normal cell, ATP is produced predominantly by oxidative phosphorylation, that is pyruvate getting converted to acetyl CoA, acetyl CoA entering into TCA cycle, and part of it, acetyl CoA enters into TCA cycle, produces reducing equivalents, which then enters ETC and then produces energy. So cancer cells cannot wait, they are rapidly proliferating, right? So they depend on lactate, pyruvate to lactates, and the ATP derived. And all the glycolytic intermediates, they serve as carbon fuels, nucleotide reserves, and amino acid reserves. So what's the answer here? So the Warburg effect makes cancer cells immortal? No. Aerobic glycolysis with lactate formation? Yes. Why? Lactate formation is increased for the reason that I said, and it is called as aerobic glycolysis because although lactate formation occurs, it is occurring in the presence of oxygen. So it is called as aerobic glycolysis with lactate formation. Okay. And does it produce more ATP? No, it produces less ATP. Decreased glucose uptake? No, the rate of glycolysis is many folds higher in cancer cells than in a normal cell. Okay, so the right answer here is aerobic glycolysis with lactate formation. Okay, so let's move on to the next question here, which is, which of the following is true about telomeres and telomerase? So now let's discuss about telomerase first. So to discuss on telomerase. Okay, so telomeres. So telomeres nothing but the ends of chromosomes. So for example, let me draw a chromosome here. So suppose this is a 3' and this is a 5' and in the other strand this is a 5' and 3'. So how does replication start? So the first process of replication is separation of these two strands, right? The parent strand has to separate. After which, what has to happen? After which, your RNA primer has to bind. So supposing, imagine this is your RNA primer. Okay, the RNA primer, the synthesis happens from 5' to 3' direction. So both the strands, RNA primer goes and binds. And what is the next step? The next step is synthesis, right? So the synthesis of the leading strand is something like this, and of the lagging strand, it happens as fragments, as Okazaki fragments, right? So let this be the 5' and 3' of the daughter strand, and let this be the 3' and 5' of the daughter strand. So what will be the next step? The next step is nothing but, yeah, cleavage of or removal of these RNA primers, and this removal is brought about by an enzyme called DNA polymerase 1, right? After which, what happens? How will your daughter strands look like? So this is your parent strand, 3' to 5', and this is the other strand, which is 5' to 3'. So how will your daughter strands look like? Your daughter strands will look something like this. Here it will be something like this, and here it will be something like this. So as you can see, which of these two is larger? The daughter strands or the parent strands? Yeah, the parent strands are larger than your daughter strands. Why? Because this particular region of the daughter strands, they are empty, there is a lacuna there because your RNA primers were removed, right? So because of the removal of the RNA primers, there is going to be something called shortening of chromosomes, isn't it? So this shortening is the one which eventually will lead to aging, fine. So this is something which is not decided in a germ cell. Okay, in a germline cell or germ cell, we do not want this thing to happen. We do not want the ends of the chromosomes to get shortened, and for this reason here we have something called telomeres. So in the parent strand, as we said, there was 3 dash and 5 dash here, and there was 5 dash and 3 dash here, right? So this, in this 3 dash end, if you see, there is something called telomeres. So telomeres are nothing but short tandem repeats. Okay, so imagine there is this sequence TTAGG here, and then TTAGG here, and TTAGG here. Okay, so let me draw an extended 5 dash. Okay, so this sequence is there. Similarly, there is this repeat sequence on the other side, other 3 dash end as well. Okay, so now in addition, what happens is there is something called telomerase. So telomerase is an enzyme. Okay, it's a protein, it's an enzyme. So that is also present over here. Okay, so this telomerase, this enzyme also has an RNA primer. Okay, let me draw the RNA primer. Okay, let me use orange color. Okay, so this is the RNA primer. So this RNA primer is complementary to these TTAGG repeat sequences. Okay, so because this is complementary, it can bind to this TTAGG and it can bring about extension of this 3 dash end. Okay, so 3 dash end, which was initially here, so this 3 dash end was initially here because of this telomerase, activity of telomerase. So this is nothing but your telomerase. Okay, so this is telomerase, this is the protein, and then this is nothing but the RNA. So this, since this is a RNA, protein, an enzyme which is a protein with RNA, it is also called as a ribonucleoprotein. Okay, so ribonucleoprotein, your telomerase is also called as a ribonucleoprotein. So because of this telomerase activity, we have a new 3 dash end, extended 3 dash end. So hence similarly we have another extended 3 dash end here also. So ultimately now it looks something like this. Okay, so we have a new 3 dash end and a 5 dash end, and then we have sorry, shouldn't be like this. Okay, so this is your 5 dash n and this is your new 3 dash n on the other side. So this looks something like this. So this is your parent strand. This thing happens before replication sets in, and now when replication sets in, what happens is that your primer has to bind, right? So now primers will bind to this extended sequence here. Okay, so this extended 3 dash ends, these are the places where your RNA primers will bind, and then eventually, as you all know, now synthesis occurs this way here and this way here. Okay, so what is the next step? Next step is a cleavage of these RNA primers, which is brought about by DNA polymerase 1. So after which, what is the how will it look like? So it will look like something like this. Okay, 3 dash n and 5 dash n. This is your 5 dash n and this is your 3 dash n of the parent strands, and your daughter strands will look something like this. So the both the parent strands and daughter strands, they are of the same length now. Okay, so and now I told you that this thing happens only in your germ cells and not in, it never happens in somatic cells. These somatic cells, they are devoid of telomerase. If telomerase are present in somatic cells, then we'll end up getting cancer, because somatic cells have to eventually die. The chromosome has to shorten and shorten, and eventually it has to die. If this is not happening, then we'll end up getting cancer, which is undecided. Okay, so that is why telomeres are not present in somatic cells, rather it is present in germ cells. Fine. So with this concept, let's move on to the question now. Okay, so this is the question. So the question here is true about telomeres. Okay, the first option here is increase telomere length with aging. So do telomeres increase in length? No, they actually decrease in length. So option B is gradually decreasing length of telomeres with aging. Yeah, of course. So this is what is right. Okay, so this is the option that is right. So this is what I was explaining to you as well. Okay, so the third option, Telomeres have coding sequences? No, they do not have coding sequences. What they have is just the repeat sequences. Okay, that's not coded. That's not a coding sequence. Telomeres is found in somatic cells? No, they are not found in somatic cells. They are only present in germ cells. Fine. So this is about fourth question. Now moving on to the last question. The last question is, when a codon is changed to stop codon, when a coding codon is changed to stop codon, then it is called what? Okay, so now let's discuss each of these options here. So to start with, frame shift mutation. So what do you mean by a frame shift mutation? So if we all know a codon is a triplet nucleotide, right? So let me draw. So something like AGG, AGA, UGG, and then UAG. This is it. Okay, so this is a codon. Okay, this is a sequence of codons. And supposing there is insertion or deletion happening. Okay, insertion or deletion happening. Let's discuss one of these, maybe deletion. Okay, so supposing this A here is deleted. Okay, so what happens? You'll get something like this: AGG, AG, and dash, UGG, and UAG. So what will happen now? Because codon is always a triplet nucleotide, this AG is empty here. The third nucleotide is empty here. So what happens is this will invite the next nucleotide in line. So this is going to recruit this U here. Okay, so now this moves here, so this is left with GG. So this GG will invite the next nucleotide in line. So this is going to invite U, and then AG will invite the next one, probably another G, and so on. So now what happens? The entire reading frame is changed. So this AGG will code for A, this, supposing this AGA, let's talk with this thing. So this AGG will code for A amino acid, and this AGA will code for B amino acid, UGG will code for C amino acid, and UAG will code for D amino acid. But now because of this this deletion that has happened here, this AGG will code for amino acid A, but this AGU will code for amino acid X. This GGU will code for amino acid Y, and AGG will code for amino acid Z. So now the entire sequence is compromised. Okay, so this is nothing but your frame shift mutation. So this is not what is given in the question. So now let's discuss nonsense mutation. Yeah, so a nonsense mutation is a mutation where a coding codon is changed into a stop codon. So what are the three stop codons that you know of? So UGA, UAG, and UAA. So these are the three stop codons. Okay, so imagine there is a sequence of codons lined up, and one of the codon is UGG. Okay, so because of some mutation, this UGG is getting converted to UGA. So UGA is a stop codon. So there is a translational arrest. So when a sequence of codons are lined up, and one of the codon, because of mutation, gets converted to a stop codon, the translation will not be complete. It will result in a truncated protein. Okay, so this translational arrest happens. So this type of mutation is called as nonsense mutation, and that's exactly the question that is given to us. So the answer here is nonsense mutation. Okay, the answer here is nonsense mutation. So just let's discuss missense mutation and silent mutation. What do you mean by missense mutation? Missense mutation is nothing but supposing there is a codon here, UGG or something like that. Okay, so this because of mutation, this results in some other codon. Okay, UAA, something like that. Okay, not UA is also a stop codon. Let me write it something. AGG. Okay, so this is getting converted to AGG. Okay, so this codon codes for amino acid X, whereas this codon codes for amino acid Y. So these two are totally unrelated amino acids. So when the two amino acids are they are not the same. Okay, the two amino acids differ because of a mutation happening, then it's called as missense mutation. So this missense mutation may be of two types. Okay, this may be conservative, and or it may be non-conservative. So what is the difference here? Conservative is when the two amino acids are of the same class. For example, consider I'll give you an example. Let X be glutamic acid. Okay, glutamate, and let Y be aspartate. Okay, so both of these are essentially polar negatively charged amino acids. So these, if glutamate is replaced by aspartate, it's not going to affect the protein in a significant way, right? So that is considered as a conservative missense mutation. Okay, supposing there is glutamate here, supposing if X is glutamate but Y is, say, valine. So what has happened here? Glutamate is a polar negatively charged amino acid, whereas valine is a non-polar amino acid. Is it acceptable? No. So this mutation is not acceptable. So what is this condition here? Glutamate goes and valine as well comes. That is nothing but your sickle cell anemia, right? Sickle cell anemia. So in the beta globin gene, there is this mutation happening. So this type of mutation is called as non-conservative missense mutation. Okay, so the last in option is silent mutation. So silent mutation is nothing but supposing there is a codon here which is AGG, and because of mutation it gets changed into AGA. Okay, so this AGG codes for arginine. Okay, and this AGA also codes for arginine, okay, by the property of something called degeneracy. Okay, so multiple codons can code for the same amino acid. Okay, that is the property of degeneracy. So if this results in such a condition, then it's called as silent mutation, because there is no clinically significant effect that is happening. The amino acid is also the same, so that's called as silent mutation. Fine. So the answer here, when a codon is changed to stop codon, then it's nothing but nonsense mutation. Okay, so thank you. So keep revising. All the best.